Imagine this. Inside a silent laboratory, billions of microscopic particles are carefully prepared to protect millions of people. Most people never think about this, but every vaccine begins with carefully selected biological materials and nutrients. Scientists work in controlled cleanrooms where every surface, tool, and ingredient must meet strict safety standards. Large bioreactors, filtration systems, and quality control laboratories all play a role in vaccine manufacturing. This process combines biology, chemistry, engineering, and precision manufacturing to create a safe vaccine. In the next few minutes, we will follow the journey from raw materials to the final sterile vial. Every vaccine starts with carefully sourced ingredients that provide the foundation for the final formulation. Purified water forms the largest component in many vaccine formulations and must meet pharmaceutical-grade standards. Salts and sugars help maintain stability and create the right environment for vaccine components. Proteins and biological materials are prepared under carefully controlled conditions to ensure consistency and safety. Before production begins, technicians test every material for purity, sterility, and precise composition. Specialized instruments can detect tiny impurities that will be invisible to the naked eye. Only materials that meet strict quality specifications are approved for the next stage of manufacturing. What happens next is where these raw materials begin transforming into a carefully controlled vaccine substance. The manufacturing process begins inside large stainless steel bioreactors designed to maintain sterile conditions. Inside these vessels, cells or biological material are grown under carefully controlled temperature and nutrient conditions. Sensors continuously monitor temperature, pH, oxygen levels, and mixing speed throughout the process. This growth phase may continue for several days until the desired biological material reaches sufficient concentration. Once the target concentration is reached, the vaccine material is harvested from the bioreactor. The harvested material then enters a purification stage designed to remove unwanted substances. Filtration systems separate the desired vaccine components from impurities and residual materials. Additional purification techniques may further concentrate and refine the active vaccine substance. Scientists test samples during purification to ensure the material remains stable and effective. After purification, the vaccine substance is formulated with additional ingredients to create the final mixture. These ingredients help stabilize the vaccine and maintain its effectiveness during storage and transport. The formulation is mixed gently to ensure the components are evenly distributed. At this stage, the vaccine mixture must remain sterile and free from contamination. Specialized cleanroom procedures help prevent particles or microorganisms from entering the product. Samples from the formulated batch are tested again before moving to the filling stage. Only batches that meet strict manufacturing standards continue to the next phase. What happens next is the critical quality control stage that determines whether the vaccine is safe for use. Quality control begins with extensive testing to verify the vaccine's safety, potency, and purity. Scientists analyze samples to confirm the active components are present in the correct amount. Sterility tests check whether any bacteria, fungi, or other microorganisms are present. Potency tests determine whether the vaccine can produce the intended immune response. If any result falls outside the required specifications, the batch is investigated further. Only batches that pass every quality test are approved for filling and packaging. The approved vaccine is transferred into a sterile filling line inside a cleanroom. Automated machines fill each vial with a precise, measured dose of vaccine. The vials are sealed immediately to maintain sterility and prevent contamination. Labels are applied to each vial, providing important information about the product and batch. The labeled vials are inspected again to ensure the seals and labels are correct. Approved vials are packaged into cartons and prepared for refrigerated storage. Many vaccines require cold chain storage to maintain their stability and effectiveness. Temperature monitoring systems continuously check storage conditions to ensure vaccine integrity. The packaged vaccines are then prepared for shipment to healthcare facilities around the world. Cold chain logistics help keep the vaccines at the required temperature during transport. At healthcare facilities, medical professionals receive and store the vaccines according to strict guidelines. The next time you see a vaccine vial, remember the years of research and precision behind it. From raw materials to sterile packaging, vaccine manufacturing combines biology, engineering, and rigorous quality control. This process represents a remarkable collaboration between scientists, engineers, technicians, and healthcare professionals. Through careful manufacturing and testing, vaccines are prepared to help protect public health worldwide.